So the first speaker I'd like to welcome back is Mark. As you know, Dr Mark Gilchrist is a specialist pharmacist who works at Imperial College, and he spoke to us this morning, which was an excellent overview, and he's going to be extending the discussion to talk about IV antimicrobial stability, evidence or evidence-free, and I understand that meropenem, our friend, may get a mention. Thank you.
Okay, thanks Darren, thanks again to the organisers and for allowing me to speak. And you're all back after the morning break, so still interested. So I've got a disclaimer that I put up when I was writing this talk, mainly because of my background and also because drug stability causes some debate, and I hope that it will cause lots of debate as we go through. I'm not proposing that we have all the answers, but what I'm going to try and do through some case studies and through some data is describe what is out there, and I would welcome the challenge from the audience. I'm sure it will go on, but I'm going to start with a case study, if that's all right, and we'll go from there. So this is a lady that came to us just before Christmas, and basically she wanted to go home before Christmas, and I want to see what you would do as an audience. So you've got a question in a minute. So this is a 71-year-old female. She'd been on holiday in Spain. She'd had right ear pain since the end of September, really with some itching. She hadn't gone swimming or done anything exotic. She'd just gone to enjoy some winter sun. She came back, she went to her GP, she got given some steroid drops. No better, so some of you are shaking your head already at steroid drops. She was referred to ENT and she got given some amoxil, which didn't have any effect, and she ended up being admitted on the 19th. So she had some intravenous antibiotics, some piptazo, and she had some reactions to that and ended up having levofloxacin and some cipro drops again and sent home. She was then readmitted. And this is all unknown to the infection service. She's then readmitted and she's given some mero with some advice from the on-call infection services. But I guess demonstrating the stretch and the reservedness of infection services, she wasn't completely followed up. Given a week of cipro and sent home. So she stops, when she goes home and she switched to oral, she gets the pain, it comes back again. She's a renal transplant patient, so she gets given some interesting antibiotics in her renal clinic. She's given some doxy because she's got an MSSA on recent culture. Pain gets worse. She gets some discharge from the ear. She then develops a nerve palsy and has an eye and mouth drop. And so she's readmitted just before Christmas on the 17th of December, and mero is started in her. So the infection team is then called. Note that we're called 4 days after she's admitted. Her CRP is a lot better. She's got no pain, fevers, no pus. Always worry about herpes and zoster type things. So actually she didn't have any of that. No confusion, no meningism. And she's having some microsuction as well. She's got some pseudomonas that's not spelt right. And she's got some MSSA. She's had some imaging, so she had some CT imaging when she came in that said no evidence of MOE, but actually you could argue that CT is probably not the best imaging for that. And she's a diabetic. She's had a renal transplant with good function, glaucoma and asthma as well. Her husband passed away a year ago, so she's keen to get home. She lives with her daughter nearby. She's got this palsy, but her other neurology is actually intact. So because of the seventh nerve palsy, because of the immunosuppression, because of type 2 diabetes, we suggest 2 weeks as a minimum and review her clinical course. We said asked for an MRI, and we did some fungal markers as well, just to rule out any fungal disease. So the question for you guys is, given this is Christmas holiday season, she doesn't want to be in hospital, she wants to get out, we're asked to send her home, what would you guys do? So you can either choose mero and ceftazidime. I'm interested to know which one you'd probably go for. So that's the first thing. So hands up if you would go meropenem. Hands up. Some of you are uncertain. Hands up if you would go ceftazidime. So more of you would go ceftazidime. Okay. And then, how many of you would use a 24-hour infuser of either agent? How many of you would use an 8-hour infuser? And how many of you would use sort of bolusing TDS? Okay, so within the audience, there's a sort of split between what you're doing, and that's pretty much what I would hope for, because one of the challenges that we have in OPAT certainly is this era of drug stability. It's one of the challenges around having not narrow spectrum agents, around collateral damage and lack of rapid administration. But one of the fourth category that we have challenges within HITH and within OPAT is certainly drug stability. And specifically, it's around the lack of validated studies. And it's the validated bit that's really important in this. Because we tend to use, as we said before, once daily broad spectrum agents, mainly because we don't have faith in the older agents. So the OPAT BSAC strategy, one of the strategy aims for the last couple of years has been to extend the range of anti-infectives that we use in an OPAT pathway by looking at standardized open access data. Okay, so we know that some of the data that is out there hasn't been validated. We know that some of the data is kept behind closed doors for various reasons. But we wanted to create an arena where we did stability testing, had an open access. It's available to anyone, so you can go and look at the data that I'm going to present, and you can drill down and look at does it support my case, does it not support, and build a business case that we put lots of agents through the program, and make it open access, as I said. So the drug stability program is on the e-opat.com website. And it was, as I say, it was really put together for local centres to use because of the restricted nature in other commercial settings, and allow for a resource for NHS care. Now in the UK, we've got a slightly, I would say, stricter pharmaceutical assurance committee. I've never been to one of these committees, but I have seen what they do, and really they're there to provide assurance to NHS hospitals around how patients can have safe injectable medicines. That's what they're designed to do. And they do that with a yellow cover document, so as it suggests, it's bright yellow. And it's there to ensure that it goes beyond the manufacturer's guidance. It tries to give some assurance into temperature, how the viscosity of an agent, the pH, the colour, even more so than the published literature. And it's got a very strict criteria. It ensures that the product that you're going to end up with, so starting and ending up with, has 95 to 105% of the starting concentration. Okay, now some of you might say, why is it over 100%? And that's because when you sometimes make up these infusers, you sometimes it allows for overage, so you sometimes have more drug than you think you do in the system. But our European and US colleagues have a slightly more relaxed view, where they go down to 90%. So the UK is strict in its nature, and I get that that's a limitation of what you're going to see. But it goes back again to this nature of trying to fulfill the best for our patients, whether it's a stewardship arena or whether it's just through safety. So, as I've said, it looks at colour, it looks at clarity, it looks at precipitation, it looks at pH, it looks at moisture loss of devices and degradation products, and are those degradation products harmful? And it also looks at the concentrations that you get in the device itself. And they test a range of temperatures, so they must be done in triplicate or a minimum duplication. And they're tested at 32 degrees, 4 time points, and they look for, does the drug have a loss factor of 5% from its initial starting concentration. Okay, so this is all very techy, but I'm illustrating this so you just understand where I'm coming from when we come to some of the results. So all of this in the UK started to give us lots of debate, and we reached out to OPAT centres across the UK and said, actually, what are you guys doing? What are you doing in practice? And we asked, what antimicrobials are you actually using? What devices are you using? What antimicrobial data is there? And do you have any local stability data? And so we asked a number of organisations, and some were nurse-led, some were patient-led, and some were specialist pharmacy units making up these drugs, either through syringes, through infusion bags, or through devices, elastomeric devices. So you see there's a range of administration going on across the UK. And when we actually started asking people, it ranged from amikacin all the way through to your beta-lactams, through to your flucloxes, your meros, TAZ, and vancomycin. And locally, it was locally validated, so they said, actually, we do this and it's fine. Okay? And that's sort of been the mantra. People look at the data, it works for them, why change something that's been working for ages? It gets people out of hospital, why would we change that? But actually, in terms of NHS organisations, you can't just transfer something that's done locally to another hospital because they want to have assurance that it's going to be okay. You can't just say, we've been doing it for years down the road, it's fine, why don't you have a go? The clinical risk, the risk-averse nature of us comes into play there. So there is a positive evidence around published evidence and reproducible evidence. So we had to go at looking at what does the literature actually show, and when you search for drug stability, going back from sort of 2013 and before, there's actually 581 articles that talked about drug stability. We took out duplicates, and that brought us down to 420, and then the final review got down to 134 titles that we could look at. Of those 134, 121 were actually accessible, and what you've got here on the left-hand side is the yellow-covered documents standard. So testing for 37 degrees, did it have the actual product inside it that it said it did? Could it be reproducible at 4 time points, etc.? And most of the data that you're seeing, that's your total down there, and not all of them were able to meet that. And certainly if you go across here, the number of devices that had 90 to 105% within their limits was 0. The drugs in question were around aminoglycosides. I didn't know this, but aminoglycosides have been the biggest infuser devices published. There were certainly a lot of them in the 1990s and early 2000s, where most of the data comes from. Beta-lactams the next phase, and then you go up. And I was surprised that quinolones were as well. So IV cipro was put in devices and done in the States. But what it meant was that within the UK, and again the strict criteria, is that there weren't any published studies that the UK could recognise for stability testing. And that started to cause the OPAT community a little bit of noise, because we didn't know if we were unique, if we were just making a big deal about this, or we were being too particular about it. So we went further afield. So there's an OPAT forum that you're very welcome to join. It's called Global Appeal. And this was our Singapore colleagues who said, well, I wouldn't recommend ampicillin. There's minimal data, and it's unstable within devices, and we just wouldn't do it. And then you have the likes of James coming along and saying, yeah, well, we do mero and ampicillin. What are you talking about? We do this all the time, and we just double doses and just get on with it. And then the MHRA in the UK starts talking about, well, we've got some latest data about ceftazidime, and we think there should be a reduction in the expiry of those because there's a potential risk of toxicity. So they reduced the shelf life of these from 10 days to 6 days, which in my practice caused me a real headache, because we use this for bronchiectatic patients. And a 6-day shelf life from it being made to arriving in the hospital means we've only got 4 days. And our bronchiectasis patients come back once a week. And I personally think it's a bit unfair to bring people back who can't really breathe that well twice a week when you could only do it once. So we stopped using ceftazidime. which I think has been a real shame for us, and so we're now using more marrow. So there's a stewardship argument on the reverse in that. And again, this was brought in because there were concerns about degradation products, but nobody has repeated the studies from a UK stringency point of view. And you've seen this before, but we've got this whole issue of AMR. So what we ended up doing is we funded 2 stability studies. One was flucloxacillin and the other is meropenem. And we used a Baxter device and we used an AccuFuser device by a different company. And we used the yellow document criteria as a way of satisfying the requirements. And what we found in the initial stability studies is that fluclox itself was very unstable in water for injection. It's very unstable in just saline if you're going to use it for a long period of time. And then the mixture of a citrate and phosphate buffer, it was unstable again. And there was high degradation rates, and also we found precipitation in the devices itself. So this was done by an independent company who won a tender from us to do fluclox and meropenem at the same time. So we came up with a 0.3 citrate buffer. So this was also chosen because it was commercially available. Because remember, the whole point of this was that NHS hospitals up and down the country, if you had a centrovenous additive service or you had a pharmacy service that could make them on site, you could do this with whatever products you had available in the UK. So this 0.3% solution is available for manufacturing services. And we used 2 concentrations of fluclox, and we wanted to make sure they were stable for 24 hours and when heated at 37 degrees. So 10 milligrams and 50 milligrams per mil concentrations we used. They were tested in triplicate. They were stored for 7 days in fridge temperatures, and then at 24 hours at 32 degrees, and then stored at 14 days at 2 to 8 degrees, and then again at sort of body temperature. And what you find is that here is what you're looking at. So you're looking at the percentage of fluclox that was remaining in the devices at 14 days and at 10 days. So it didn't change very much from the initial concentration, so it was within the 95%, and again in the Baxter LV10 devices. So what you find is that in both of the devices chosen in a 0.3 citrate buffer, that fluclox was stable. And then we came on to do meropenem. So this was the next one that was asked, what's the influence of buffering and does it meet the challenges of the yellow cover document itself? So we know that meropenem is unstable. We know that it's affected by pH, and we know it's affected by that affects the drug concentrations. And when we tested it in the studies, so exactly the same methodologies as the fluclox studies, buffering actually made no noticeable difference to the concentrations and to the end product. We knew that whether it was high or low, you had an instability. And in some devices, you had up to 50% loss after 24 hours. So this is where the argument comes around of, actually in practice, do we just double the dose and not worry about it? But you could argue that's not necessarily good stewardship. And then interestingly, whilst most studies have been carried out with a pH of 6 in these buffers, we found that a pH of 6 actually increased the degradation rate. So we went for a pH of 7, and what we ended up finding was this. So green here is your saline, the blue is the buffered 1%, and then buffered again 5% citrate. And if you take the 90% level, sort of being here, that's the European, 95% is slightly higher, so that's the UK. You find that after about 6 hours, we weren't able to validate any data past that time. So going on to 24 hours in here, we were losing up to about 40-50% in some of the devices. So that's where our argument came from, to say actually we can't find under the UK criteria this sitting definitely not 24 hours, but we postulated at 6 hours. It would be that's what we'd recommend. Now there's a slight difference in this. There are studies that are out there that look at if you compare them to the European model, so the 90% fraction of drug, that 8 hours is recommended. So that would be here. And it is all to do with the semantics of where this percentage cutoff is. And certainly we're very open to the idea of trying to get global consensus on this, because it's absolutely bonkers, in my view, that the UK has a very strict contingent. Our European colleagues across the channel, for the time being, we're still in Europe, but we may not be. And then you have the US doing something different, and Australia doing something different. And we are putting a call out to have some key opinion leaders from all of the countries come together and sit down and go, actually, we need consensus. Because there's no point in doing these analysis. The analysis to do infusions takes time, it takes money. It takes a lot of money to do these studies. And if we're saying 6 hours based on our 90% rule, whereas if you go to France, they do 8 hours based on a 90% rule, there must be some in between. But what people have not been able to demonstrate yet, in clinical trial practice anyway, is what's the effect of actually, what's the negative effect, doubling the dose and just seeing how you go, which is the argument that always comes back. And again, this is exactly, I forewarned James that I was going to put this up, but we start tweeting about this, and then halfway across the world someone says, well actually if you're a pragmatist, you just double the dose, don't you? And then some more cynical views around it doesn't get to 32 degrees in the UK anyway, so what are you worrying about? So that's fine, but the whole premise of this is to try and get some consensus. And there is data around for every, if you hold meropenem in a fridge, so 2 to 4 degrees, 2 to 5 degrees, for every day it's there, it will degrade by 1% of its total value. So if you have it in there for 14 days before you give your patient their last dose, the potential of the whole product to degrade by 14% is there. Okay? So whether that has any reality in practice is slightly different, but that's the debate I'm sure we're going to have. If you look at the other data sets across the world, Australia absolutely has produced some data. Singapore has. The Swiss are the new up-and-coming folk in this arena, as well as France. So there's 4 papers that I've put on the board there that look at amoxil, mero, vanc, and clindamycin. So in France, they did clindamycin. So we know about certainly vancomycin's use, particularly in ICUs and in other settings. Amoxil and meropenem, which we were going away from in the UK, but there's certainly data up there. And they all said that there was relative success in their patient populations, but there may possibly be the need for therapeutic drug monitoring of these agents to see whether or not it's actually having an effect. The Swiss then went on to do Fluclox, cefepime, vanc, and piptazo, and they did quite robust studies looking at drug concentrations, antibiotic exposures, and actually said they observed a high rate of success in all of their patients. Okay, and that then brings you on to saying, well, actually, what's the impact of buffering? You know, what does it do? Does it have any effect on human physiology? And what's the effect of using repeated buffers? So there's been some local studies done, certainly in the UK, where they looked at patients who'd undergone continuous infusions of beta-lactams. And the reasons are on the screen there around actually, you know, does it have an effect on your potassium, your sodium, your adjusted calcium, your INR, and your bicarbonate? And there are effects that you, when you're using buffered agents, particularly in ICU, particularly in other settings, that it has an effect. But does it have a detrimental effect? And that's really the question there. So they took 11 patients and they looked at Fluclox continuous infusions and piptazo continuous infusions for the pathologies that are on the screen there. They did extended bloods once the patients had stopped. They made sure they did them once a week, and then after they'd stopped. And they did notice some minor changes in some of those electrolytes, those falling below normal limits and those sometimes above normal limits. But in no patient was the continuous infusions interrupted, and 9 out of those 11 patients were cured or improved, and no failures were due to buffers. But it's something that is out there. And then certainly the Australian HITH community have looked at temperatures. So this was a study that looked at temperatures, whether you put the device under the duvet or over the duvet when you go to sleep. which I had not even thought of before. But does that make a difference? And it makes a difference to flow rates. That's what it's sort of looking at. And again, the Swiss then come back at you saying, well, actually, does it really make that much of a difference in practice? So these are the drugs. They've tested Fluclox, Cefepime, Taz, piptazo, and actually in between the times at 0, 12, and 24, they found it was very unlikely that there'd be insufficient antimicrobial coverage. So the data is coming and it's emerging. The IDSA guidelines have come out, but again, if you read the small print, there's caveats. So devices, there's very little published information about these. That it's they're very good. IDSA going, actually, we're not going to give any opinion. We're going to just leave it to local folk to make up their own opinion. But they do mention about stability of agents. So they mention that some drugs may have to be reconstituted in the home via very techy devices. So the mix comes a bit like a James Bond movie. You know, the mixture of antibiotic and saline happens at the point of delivery rather than it being stored in a fridge. So for us anyway, piptazo is definitely on the horizon. We started testing that 2 weeks ago. So the data for that is going to be published in an open journal as well. Ceftazidime, we started doing that a week ago. And we're really looking at the instability agents and the pyridine formation. And we've also got a grant from MSD to look at ceftolozane tazobactam in the devices as well. And again, remembering these are for UK guidance, and I keep going back to the point that it's bonkers that we're doing these things in isolation when all of us are experiencing problems with stability and the impact that has to our patients on the ground. So the ceftazidime debate is interesting. So for a long time it's been used in critical care, and for a long time it's actually been used in cystic fibrosis, using higher concentrations than we've probably used within routine practice at a clinical level. So there's various studies that have looked at 4, 22 and 33 degree temperatures. And the EMA set a limit for degradation of ceftazidime to 2 milligrams per day for pyridine as a limit, using unbuffered solutions of ceftazidime. And if you look at that paper, and again you go with a 95% limit, you start to see that even if you use some of the higher concentrations, that after 24 hours you get a significant level of pyridine formation in the infusers. And I look at this being I was a regular user of ceftazidime infusers, and they seemed to work. So I didn't get patients with neurological symptoms. So there is a degree of how we interpret this data, and we moved from 24-hour and we moved to sort of 2-hour infusers, and now we stopped using them based out of practical aspects. And again, even when you drop to 6 grams in the ceftazidime infusers, and you still get a degree of pyridine formation. The question is, does that have a clinical significance at the end? The ceftolozane tazobactam has been published, so there are some published data out there, and I'm aware of other studies that are ongoing or have been done. But again, you look at the small print. No duplication of testing was done. It was done at ambient room temperature, and if you were in Europe or here or the US, you would get away with it. But in the UK, we wouldn't have accepted that level. So we've got a study ongoing. It's looking at 2 elastomeric devices. It's looking at 4.5 grams, and again, it will be open access and as per the NHS yellow cover document. What we have done is the yellow cover documents tried to be amenable to the OPAT community. So this is available online if you want to look at it. It's trying to give OPAT services the benefit of the doubt when it comes to stability testing. And it tries to advise clinicians or pharmacists or nursing staff around drug stability, including how you might design studies, use of buffers, compounding, procurement and things. So that's available to the OPAT community as a result of some of the work that we've done. But The OPAT community is asking for, certainly from the UK, benzyl penicillin and amoxil data to go through the same process, cefepime and temacillin. And the argument here with temacillin that you can get in Europe fairly readily is that they've done all the testing. They've put it through infusers. They've put it through what they feel is adequate. But we have to work in partnership with them because the UK has a strict 95% degradation product limit. And it's how we work with those people and how we work with industry to manage the expectations of the OPAT community. As I said to you before, I think there needs to be global consensus. I think there needs to be everybody coming together and saying, okay, well, what do we do in the UK versus what do we do in the States, in Asia, in Australasia, and coming together with one global consensus that manufacturers can work towards, but also looking at what happens to patients at the end of the day. What happens to our patients that get ceftazidime? And answers James's point about just doubling the dose and getting on with it. And there is a bit about that, because how do you talk to your patients around saying, well, actually, we were giving you a drug that we can't give you anymore because legislation says we can't. And that puts people in a really tricky position, because you start doing something that's different to legislation. We do it all the time, I guess you could argue, with antibiotics. But hospitals get a bit risk averse. And it's how you manage those expectations and are able to talk to that creates a challenge. So BSAC is looking to form this global group, so watch out. We may be coming to people near you to help advise. And again, in a very open way, and we're talking with commercial partners about how we might do that to really help satisfy the needs of the global OPAT HITH community. So I guess, seeing some of the data that I've presented to you, I won't ask for a show of hands now, but would you answer any differently to how you answered at the start about what devices and things you would use in the management of those patients? I'll leave it to your conscience to decide. Those behind the scenes that do a lot of the work that can't be here, say Connor and Tim in the top there, do a lot of the stability work, and then again everyone within the program who contributes to various arms of it. And if you want to know more about it, there is as I said before, a drug stability testing tab on the OPAT website, and all of this is open access, as well as the posters that are presented at conferences and some of the international stuff. And what we're going to try and do, if the global audience warrants it, is we're going to create an online repository for everyone globally to access some local data. So if you've got data that says that stability is okay, there's going to be a portal that you can upload that data to and share it with the rest of the global community. We won't be able to validate it as a society, but it will be there as a cross-reference to save you having to go and search the PubMed literature for it all. So that's new and it's coming, and it will be here in a month or two. But thanks very much.